now the health effect of GMO foods are unknown, and because of that, some people try to stay away from these foods. Another reason is that some manufacturers try to not mention on labeling that the foods are made or developed by genetic manipulation because that would affect their business. And lastly, the high price. Everyone knows anything that has a non-GMO label is more pricier than the one that is GMO. So our question was, as a customer, can I trust on the non-GMO labeling on the foods or products? And because of the high percent of modification in soybeans, we hypothesized that the non-GMO soy protein powders that we were testing do contain the GMO ingredients.
So to test our hypothesis, the materials that we use were the whole food dried Roundup Ready soybeans, the non-GMO soybeans, and for our negative non-soy controls, we used wild rice and lentils. And for our actual protein powder samples, we used 3 different versions. They were all labeled non-GMO. We had the GNC Superfood Soy Protein Powder, the Bob's Red Mill Soy Protein Powder, and the organic natural nutraceutical soy protein powder. We chose those samples because the protein shakes have become so popular that this day and age a lot of people are drinking them, and therefore they're getting that strong dose of soybeans. And in addition to that, soybeans is present in so many of our average of everyday products. So we thought that this would be a good representation of the soy that is out there. For the soy-specific lectin detection, we started with DNA extraction, and then we tested those products on the nanodrop where we checked concentration and purity. We then were able to use those products on the PCR machine using that particular temperature for denaturing, annealing, and elongation. The primers that we used were the GMO primers 3, which was our forward, showing the sequence on the top, and the GMO primer 4, which was the reverse, shown on the bottom. And we were then able to visualize the detection of the soy-specific lectin on gel electrophoresis, looking for the band specifically on the product size of 118 base pairs. And these are our nanodrop results. For concentration, the acceptable range is anything above 1, and the purity acceptance range is between 1.6 and 2.0. As you can see, all of our samples came very close to that or within that range.
Okay, so as Teresa said, we were detecting for lectin in our samples. This lectin gene is soy specific, so we wanted to make sure that the protein powders first that we chose were indeed soy based. So as you can see, all of the samples showed up, it's kind of faint on here, but showed up bands around 118 as she mentioned, and this is the soy specific lectin gene. We did use both a KD ladder, 1 KD plus ladder, and a low range ladder just to make sure that if we did get any bands, they wouldn't fall either at the top or bottom, hopefully around 118, which they did, except for lentils, because lentils is not soy. So after we confirmed that we had soy-based products, we went on to ELISA. So we actually did a double antibody sandwich ELISA, or DAS ELISA. We first prepared our samples, and then we went through our ELISA protocol. We then collected the data using these biotech plate readers, and this machine works by optical density, so the color change in our samples would indicate positive or negative results, and it turns those colors into numerical values that can then be analyzed, which we did in Excel. So for ELISA, for a positive result, we needed a value greater than 0.3, and for a negative result, we needed less than 0.1. Anything that fell within that range between 0.1 and 0.3 was actually indeterminate. So for our Roundup Ready and our internal positive control, both of them came came out with positive result as expected. They were well above 0.3. And then our other protein samples, all the protein samples that we tested, as well as our non-GMO, came out as negative results, indicating that there's no evidence of the genetic modification in these non-GMO labeled soy protein powders. And again, this is just another chart of the data. The average values is what was used in the chart, as well as the standard deviation as the error bars. However, the p-values were at 0.05 significance. Anything low indicates that there is a significant difference, which makes sense because we did the Roundup Ready versus non-GMO, Roundup Ready versus each one of our samples, indicating again that there is a significant difference between the obvious genetic modified ones and the hopefully non-genetically modified ones.
All right, and to kind of just sum everything up for our experiment, since a large amount of soybeans are actually genetically modified in the States, we wanted to kind of verify these non-GMO labels on these soy-based protein powders. So we originally hypothesized that the 3 protein powders that we chose were going to be in fact genetically modified, but as you can see, that was not the case. So we started off with a PCR gel electrophoresis to detect the lectin gene, which would tell us that these soy-based products were actually soy, which they were. And then we went ahead and did our ELISA, and our ELISA results told us that there was no detection of protein of the EPSPS4 gene, telling us that they were actually not GMO. They were right, the labels were right, they were not genetically modified. So further experiments that we could go about from this would be maybe to repeat the experiment again to verify our results. Maybe use different soy-based protein powders, or even just soy-based anything that are labeled non-GMO. Maybe we could also test the presence for the GMO gene, the GMO gene being the EPSPS4, using PCR gel electrophoresis. And then the next one would probably be to conduct a real-time, aka RT-PCR, to detect EPSPS4 gene expression. So in conclusion, the significance of our experiment was to provide the public with scientific data that what they were buying were in fact non-GMO and not GMO, that the labeling was correct. According to our gel electrophoresis, our PCR, we did find out that our soy-based protein powders were soy, and according to our ELISA results, that these proteins of the EPSPS4 were not being produced in these soy-based products. So consumers can be confident in purchasing these products that we have conducted this experiment on.